Chapter 9. Biomolecules. 1. To find the composition of elements in living tissue, we take a liver and grind it in dash using a mortar or pestle to obtain a slurry. Trichloroacetic acid. Trichloroacetic acid. Trichloroacetic acid. 2. When we strain the slurry through a dash or dash, we would obtain two fractions. Cheesecloth or cotton. Cheesecloth or cotton. Cheesecloth or cotton. 3. How can we know that the living tissue also has inorganic compounds? By burning. Burning the living tissue will oxidize all the carbon compounds, and what will remain will be elements like Ca, Fe, etc. By burning. Burning the living tissue will oxidize all the carbon compounds, and what will remain will be elements like Ca, Fe, etc. By burning. Burning the living tissue will oxidize all the carbon compounds, and what will remain will be elements like Ca, Fe, etc. 4. PO43 and S042 are some inorganic constituents of living tissue. True. 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 5. Elements which are present in more percent in the human body than in the earth crust are 5. CHONS, carbon, hydrogen, oxygen, nitrogen, sulfur. CHONS, carbon, hydrogen, oxygen, nitrogen, sulfur. CHONS, carbon, hydrogen, oxygen, nitrogen, sulfur. 6. Identify the organic compounds found in living tissues shown in Dig Arc. I. NEET. A. Alanine, B. Serine, C. Glycine, D. Uracil, E. Adenine, F. Thymine, G. Guanine, H. Cytosine, I. Phospholipid lecithin. A. Alanine, B. Serine, C. Glycine, D. Uracil, E. Adenine, F. Thymine, G. Guanine, H. Cytosine, I. Phospholipid lecithin. A. Alanine, B. Serine, C. Glycine, D. Uracil, E. Adenine, F. Thymine, G. Guanine, H. Cytosine, I. Phospholipid lecithin. 7. Amino acids are substituted dash. Methane, methane, methane. 8. The four substituent in an amino acid are hydrogen, carboxyl group, amino group, and a variable group designated as the R group. Hydrogen, carboxyl group, amino group, and a variable group designated as the R group. Hydrogen, carboxyl group, amino group, and a variable group designated as the R group. 9. How many types of amino acids occur in proteins? 20. Dash. 20. Dash. 20. 10. Amino acid name when R group is methyl. Alanine. Alanine. Alanine 11. Amino acid name when R group is hydroxymethyl. Serine. 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 12. Amino acid name when R group is hydrogen. Glycine. 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 13. Acidic amino acid X. Glutamate, aspartate. Glutamate, aspartate. Glutamate, aspartate. 14. Basic amino acids X. Lysine, arginine, mnemonic, lyre. Lysine, arginine, mnemonic, lyre. Lysine, arginine, mnemonic, lyre. 15. Neutral amino acid X. Valine, glycine, alanine, leucine, isoleucine, mnemonic, Vogeli. Valine, glycine, alanine, leucine, isoleucine, mnemonic, Vogeli. Valine, glycine, alanine, leucine, isoleucine, mnemonic, Vogeli. 16. Aromatic amino acid X. Tyrosine, phenylalanine, tryptophan. Tyrosine, phenylalanine, tryptophan. Tyrosine, phenylalanine, tryptophan. 17. Switter means dash. This property is present in hermaphrodite, amino acid. Hermaphrodite, amino acid. Hermaphrodite, amino acid. 18. A fatty acid has a dash group attached to an R group. Carboxyl. 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 19. Palmitic acid has dash no of carbons excluding the carboxyl group. 15. Including the carboxyl group is 16. Dash. 15. Including the carboxyl group is 16. Dash. 15. Including the carboxyl group is 16. 20. 20 carbons are present in dash acid. Arachidonic. 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 21. Unsaturated fats mean have one or more C equals C bond. Have one or more C equals C bond. Have one or more C equals C bond. 22. Glycerol is chemically dash. Trihydroxypropane. 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 23. Gingerly oil has a high melting point. False. 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 24. X of phospholipids, dash. Phosphatidylcholine, lecithin, phosphatidylthanolamine. Phosphatidylcholine, lecithin, phosphatidylthanolamine. Phosphatidylcholine, lecithin, phosphatidylthanolamine. 25. Fats and oils are distinguished based on melting point. Fat has a high melting point while oil has low Melting point, fat has a high melting point while oil has low. Melting point, fat has a high melting point while oil has low. 26. Phospholipids are found in dash. Cell membrane. Cell membrane. Cell membrane. 27. Neural tissues have more complex lipids like dash, which have instead of glycerol as a backbone. Sphingolipids, sphingosine. Sphingolipids, sphingosine. Sphingolipids, sphingosine. 28. Sphingomyelin is a type of dash which consists of dash as a head group. Sphingolipid, phosphatidylphenolamine. Sphingolipid, phosphatidylphenolamine. Sphingolipid, phosphatidylphenolamine. 29. Nitrogenous bases when attached to sugar form dash, and when attached also to dash form nucleoside, phosphate. Nucleoside, phosphate. Nucleoside, phosphate. 30. Nucleoside version of adenine is called adenosine. 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 31. Nucleoside version of cytosine is called cytidine. 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 32. Nucleoside version of thymine is called thymidine. 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 33. Nucleoside version of uracil is called uridine. 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 34. Nucleotide version of cytosine is called cytodylic acid. Cytodylic acid. Cytodylic acid. 35. Flavonoids are primary metabolites. False. 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 36. Types of secondary metabolites are alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. Alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. Alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. 37. Example of pigments. Carotenoids, anthocyanins. Carotenoids, anthocyanins. Carotenoids, anthocyanins. 38. Example of alkaloids. Morphine, codeine. Morphine, codeine. Morphine, codeine. 39. X. Of terpenoids. Monoterpenes, diterpenes. Monoterpenes, diterpenes. Monoterpenes, diterpenes. 40. X. Of essential oils. Turpentine, eucalyptus oil, lemon oil. Turpentine, eucalyptus oil, lemon oil. Turpentine, eucalyptus oil, lemon oil. 41. Curcumin is a dash type of secondary metabolite. Pigment. 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 42. X. Of toxins. Abrin, ricin. Dash. Abrin, ricin. Abrin, ricin. 43. Concanavalin A is a dash. Lectins. 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 44. X. Of drugs. Vinblastin, curcumin. Vinblastin, curcumin. Vinblastin, curcumin. 45. X. Of polymeric substance. Rubber, gums, cellulose. Rubber, gums, cellulose. Rubber, gums, cellulose. 46. No secondary metabolites have ecological importance. False. 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 47. Compounds found in the acid-soluble pool have molecular weights ranging from dash to dash daltons. 18 to 800. Dash. 18 to 800. Dash. 18 to 800. 48. What are the compounds in the acid-insoluble pool? Proteins, nucleic acids, polysaccharides, and lipids. Proteins, nucleic acids, polysaccharides, and lipids. Proteins, nucleic acids, polysaccharides, and lipids. 49. Define micromolecules and macromolecules. Micromolecules, MW, less than 1000 daltons, macromolecules, found in the acid-insoluble pool. Micromolecules, MW, less than 1000 daltons, macromolecules, found in the acid-insoluble pool. Micromolecules, MW, less than 1000 daltons, macromolecules, found in the acid-insoluble pool. 50. Acid-insoluble pool has a molecular weight in the range of dash or above 10,000 daltons. 10,000 daltons. 10,000 daltons. 51. Lipids are not strictly macromolecules or micromolecules. Macromolecules. 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 52. The acid-soluble pool represents roughly the composition of dash. Cytoplasm. 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 53. The macromolecules from cytoplasm and organelles become the acid-insoluble fraction. True. 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 54. The most abundant chemical in living organisms is water. 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 55. Dash is 10 to 15% of the total cell mass. 56. Carbohydrates are dash percent of the total cell mass. 57. Lipids are dash percent of the total cell mass. 58. Nucleic acids are dash percent of the total cell mass. 59. Water percent in the cell is protein, polysaccharide, nucleic acid 60. Protein is a homopolymer or heteropolymer. 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 61. What is the difference between homopolymer and heteropolymer? Homopolymers have the same monomer, but heteropolymers have different types of monomers. Homopolymers have the same monomer, but heteropolymers have different types of monomers. Homopolymers have the same monomer, but heteropolymers have different types of monomers. 62. Define essential amino acids. Amino acids which cannot be synthesized by our body hence have to be taken from outside. Amino acids which cannot be synthesized by our body hence have to be taken from outside. Amino acids which cannot be synthesized by our body hence have to be taken from outside. 63. Name all the essential amino acids. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, arginine, and valine. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, arginine, and valine. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, arginine, and valine. 64. Function of GLUT4 is enables glucose transport into cells. Enables glucose transport into cells. Enables glucose transport into cells. 65. Dash function is an intercellular ground substance. Collagen. 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 66. Most abundant protein in the animal world is collagen. 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 67. Most abundant protein in the whole biosphere is rubisco. 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 68. Polysaccharide is in acid-soluble or acid-insoluble pool. False. 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 69. Cellulose is a polymer of glucose. 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 70. Inulin is a polymer of fructose. 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 71. Dash is a storehouse of energy in plant tissues. Starch. 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 72. Polysaccharide which stores energy in animals. Glycogen. 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 73. In a polysaccharide chain, the right end is called the reducing or non-reducing end, and the left end is called dash end. Reducing, trig, R for R, non-reducing. Reducing, trig, R for R, non-reducing. Reducing, trig, R for R, non-reducing. 74. Cellulose forms helical secondary structures. False. 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 75. Starch hold the two molecules in the dash. Helical structure. Helical structure. Helical structure. 76. The starch 12 is dash in color. Blue. Blue. Dash. Blue. 77. Exoskeleton of arthropods has a complex polysaccharide called chitin. 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 78. X of amino sugars. N-acetylglucosamine, N-acetylgalactosamine. N-acetylglucosamine, N-acetylgalactosamine. N-acetylglucosamine, N-acetylgalactosamine. 79. Complex polysaccharides are mostly homopolymers or heteropolymers. Homopolymers. 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 80. Chitin is a homo or heteropolysaccharide of homopolysaccharide, N-acetylglucosamine, NAG. Homopolysaccharide, N-acetylglucosamine, NAG. Homopolysaccharide, N-acetylglucosamine, NAG. 81. Nucleic acid is found in acid-soluble or acid-insoluble pool. Acid-insoluble. 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 82. Building block of nucleic acid is nucleotides. 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 83. Components of nucleotides are nitrogenous base, monosaccharide, phosphate. Nitrogenous base, monosaccharide, phosphate. Nitrogenous base, monosaccharide, phosphate. 84. A and G are substituted dash. Purines. 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 85. In deoxyribose, the oxygen is removed from dash position. 2. Dash. 2. Dash. 2. 86. Nitrogenous bases which are substituted pyrimidine. Cytosine, thymine, uracil. Cytosine, thymine, uracil. Cytosine, thymine, uracil. 87. Identify the various levels of protein structures shown in diagram. 2. Label P, Q, R, S and true. Diagram. 2. A. Primary. P. Polypeptide. B. Secondary. Q. Alpha helix. R. Beta platted sheet. C. Tertiary. S. Hydrogen bond. True. Disulfide bond. D. Quaternary. Diagram. 2. A. Primary. P. Polypeptide. B. Secondary. Q. Alpha helix. R. Beta platted sheet. C. Tertiary. S. Hydrogen bond. True. Disulfide bond. D. Quaternary. Diagram. 2. A. Primary. P. Polypeptide. B. Secondary. Q. Alpha helix. R. Beta-platted sheet. C. Tertiary. S. Hydrogen bond. True. Disulfide bond. D. Quaternary. 88. The sequence of amino acids, i.e., the positional information in a protein, is given in dash structure. Dash. Primary. 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 89. Right end is represented as a first or last amino acid in the left end by dash amino acid. Last, first. Last, first. Last, first. 90. N-terminal amino acid is the first or last amino acid. First, first, first. 91. Last amino acid is called dash terminal amino acid. C. 92. In protein both left and right-handed helices are observed. False, only right-handed. False, only right-handed. False, only right-handed. 93. Secondary structures X. Alpha helix, beta-pleated sheets. Alpha helix, beta-pleated sheets. Alpha helix, beta-pleated sheets. 94. The long protein chain is also folded upon itself like a hollow woolen ball, giving rise to dash structure. Dash. Tertiary structure. Tertiary structure. Tertiary structure. 95. Tertiary structure gives us a 2D or 3D view of a protein. 3D. 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 96. Dash structure is absolutely necessary for many biological activities of protein. Tertiary structure. Tertiary structure. Tertiary structure. 97. The manner in which individual folded polypeptides or subunits are arranged with respect to each other is the architecture of a protein, otherwise called dash structure of a protein. Quaternary. 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 98. Adult hemoglobin consists of how many subunits? 4. Dash. 4. Dash. 4. 99. Name the subunits of hemoglobin: A subunit and B subunit, A subunit and B subunit, A subunit and B subunit. 100. X of tertiary structure. Dash. Myoglobin. 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 101. X of quaternary structure. Hemoglobin. 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 102. No of peptide bond present in a tripeptide is 2. Dash. 2. Dash. 2. 103. In a polysaccharide, the individual monosaccharides are linked by a dash bond. Glycosidic. 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 104. In a nucleic acid, dash links the 3 carbon of one sugar of one nucleotide to the 5 carbon of the sugar of the succeeding nucleotide. Phosphate. 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 105. In nucleotides, the bond between phosphate and hydroxyl group of sugar is a dash bond. Ester. 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 106. Phosphoester bond is present in DNA. Dash. True. 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 107. Dash model exhibit one of the secondary structure of DNA. Watson-Crick. 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 108. The two strands of polynucleotides are parallel. False, anti-parallel. False, anti-parallel. False, anti-parallel. 109. The backbone is formed by sugar-phosphate sugar chain, sugar-phosphate sugar chain, sugar-phosphate sugar chain. 110. No of H bond between A and T is 3. Dash. 3. Dash. 3. 111. No of H bond between G and C is 2. Dash. 2. Dash. 2. 112% of T in RNA if percent of C is 40%. 0% RNA have uracil instead of thymine. 0% RNA have uracil instead of thymine. 0% RNA have uracil instead of thymine. 113% of A in DNA if percent of G is 30%. 20%. Dash. 20%. Dash. 20%. 114. When we methylate dash thymine is formed. Uracil, thymine is 5-methyl uracil. Uracil, thymine is 5-methyl uracil. Uracil, thymine is 5-methyl uracil. 115. In bDNA, at each step, the strand turns dash degrees. 36 degrees. Dash. 36 degrees. Dash. 36 degrees. 116. One full turn of the helical strand would involve dash base pairs. 10. Dash. 10. Dash. 10. 117. The pitch of bDNA is dash nanometer. 3.4. Nanometer. Dash. 3.4. Nanometer. Dash. 3.4. Nanometer. 118. 6. Base pair long DNA have dash nanometer length. 2.04 nanometers. Dash. 2.04 nanometers. Dash. 2.04 nanometers. Enzymes 119. Identify A, B, X, Y and Z in the graph shown in diagram. 3. Diagram. 3. X. Substrate. Y. Transition state. Z. Product. A. Activation energy without enzyme. B. Activation energy with enzyme. Diagram. 3. X. Substrate. Y. Transition state. Z. Product. A. Activation energy without enzyme. B. Activation energy with enzyme. Diagram. 3. X. Substrate. Y. Transition state. Z. Product. A. Activation energy without enzyme. B. Activation energy with enzyme. 120. Enzymes through their dash catalyze reactions at a high rate. Active site. Active site. Active site. 121. Non-proteinaceous enzyme X. Ribozyme. 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 122. One can depict an enzyme by a line diagram. True. 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 123. Rate can also be called velocity if the direction is specified. Dash. True. 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 124. Difference between inorganic catalyst and enzymes. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures. 125. Enzymes isolated from organisms who normally live under extremely high temperatures denature at high temperatures. False. 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 126. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. True. 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 127. A general rule is, rate doubles for every dash increase. 10. Dash. 10. Dash. 10. 128. Enzyme used to convert water and CO2 to H2CO3 is carbonic anhydrase. Carbonic anhydrase. Carbonic anhydrase. 129. The S-complex formation is a transient or non-transient phenomenon. Transient. 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 130. All other intermediate structural states are unstable. True. 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 131. How does enzymes bring such high rates of chemical conversions? By reducing the activation energy needed. By reducing the activation energy needed. By reducing the activation energy needed. 132. What is the catalytic cycle of enzyme action? A plus SS8, A plus P. A plus SS8, A plus P. A plus SS8, A plus P. 133. The activity of an enzyme can be affected by a change in the dash conditions which can alter the dash structure of protein. Tertiary. 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 134. What does the y-axis represent in the graphs given in diagram? 4. Enzyme activity. Enzyme activity. Dash. Enzyme activity. 135. The graph given in diagram 5 shows the effect of change in concentration of substrate on enzyme activity. Identify X, Y and Z. Diagram 5. X, Vmax. Y, Vmax are 2, Z, Km. Diagram 5. X, Vmax. Y, Vmax are 2, Z, Km. Diagram 5. X, Vmax. Y, Vmax are 2, Z, Km. 136. Factors affecting enzyme activity are temperature, pH, concentration of substrate, inhibitors, temperature, pH, concentration of substrate, inhibitors, temperature, pH, concentration of substrate, inhibitors. 137. Low temp preserves the enzyme in a permanently inactive state. False, in a temporarily inactive state. False, in a temporarily inactive state. False, in a temporarily inactive state. 138. With increase in substrate concentration, velocity of the enzymatic reaction rises always. False, do not rise after attaining Vmax. False, do not rise after attaining Vmax. False, do not rise after attaining Vmax. 139. When the inhibitor closely resembles the substrate in its molecular structure, it is known as dash inhibitor. Competitive. 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 140. Competitive inhibitors are often used in the control of bacterial pathogens. True. 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 141. X of competitive inhibition. Inhibition of succinic dehydrogenase by malonate, sulfa drugs for inhibition of folic acid synthesis in bacteria. Inhibition of succinic dehydrogenase by malonate, sulfa drugs for inhibition of folic acid synthesis in bacteria. Inhibition of succinic dehydrogenase by malonate, sulfa drugs for inhibition of folic acid synthesis in bacteria. 142. Enzymes are divided into -no of classes each with -no of subclasses and named accordingly by a digit number. 6, 10 to 13, 4. 6, 10 to 13, 4. 6, 10 to 13, 4. 143. Name the 6 classes of enzymes with their example: I. Oxidorictases or dehydrogenases, X. Lactate dehydrogenase, E. Transferases, X. Transaminase, kinase, E. Hydrolases, X. Amylase, lipase, etc., IV. Lyase, X. Aldolase, carbonic anhydrase, V. Isomerase, X. Phosphoglucose isomerase, V. Ligase, X. RUBP carboxylase, PEP carboxylase, I. Oxidorictases or dehydrogenases, X. Lactate dehydrogenase, E. Transferases, X. Transaminase, kinase, E. Hydrolases, X. Amylase, lipase, etc., IV. Lyase, X. Aldolase, carbonic anhydrase, V. Isomerase, X. Phosphoglucose isomerase, V. Ligase, X. RUBP carboxylase, PEP carboxylase, I. Oxidorictases or dehydrogenases, X. Lactate dehydrogenase, E. Transferases, X. Transaminase, kinase, E. Hydrolases, X. Amylase, lipase, etc., IV. Lyase, X. Aldolase, carbonic anhydrase, V. Isomerase, X. Phosphoglucose isomerase, V. Ligase, X. RUBP carboxylase, PEP carboxylase. 144. Enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis leaving double bonds are called lyases, lyases, lyases. 145. Enzymes catalyzing the linking together of two compounds are called ligases. 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 146. Define transferases. Enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S. Enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S. Enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S. 147. Define isomerases. Includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. Includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. Includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. 148. What are cofactors? Cofactors are non-protein constituents bound to the enzyme to make the enzyme catalytically active. Cofactors are non-protein constituents bound to the enzyme to make the enzyme catalytically active. Cofactors are non-protein constituents bound to the enzyme to make the enzyme catalytically active. 149. What is apoenzyme? The protein portion of the enzyme. The protein portion of the enzyme. The protein portion of the enzyme. 150. Name the 3 types of cofactors. Prosthetic group, coenzymes, metal alkenes. Prosthetic group, coenzymes, metal alkenes. Prosthetic group, coenzymes, metal alkenes. 151. Dash are organic or inorganic compounds and are tightly bound to the apoenzyme. Prosthetic group, organic. Prosthetic group, organic. Prosthetic group, organic. 152. Dash is the prosthetic group in peroxidase and catalase. Heme, heme, heme. 153. Function of peroxidase and catalase is breakdown of H2O2 to water and oxygen, breakdown of H2O2 to water and oxygen, breakdown of H2O2 to water and oxygen. 154. Dash are organic or inorganic compounds but association with apoenzyme is only transient. Coenzymes, coenzymes, coenzymes. 155. X of coenzymes. NAD, NADP, NAD, NADP, NAD, NADP. 156. The essential chemical components of many coenzymes are dash. Vitamins, X, niacin B3. Vitamins, X, niacin B3. Vitamins, X, niacin B3. 157. Metal alkenes form dash bonds with dash at the active site and at the same time form one or more coordination bonds with the dash, coordinate bonds, side chains, substrate, coordinate bonds, side chains, substrate, coordinate bonds, side chains, substrate. 158. Zinc is a cofactor of carboxypeptidase, carbonic anhydrase, carboxypeptidase, carbonic anhydrase, carboxypeptidase, carbonic anhydrase. 159. Maltose formula. C12H22O11. 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 160. Catalytic activity is lost when the cofactor is removed from the enzyme. True, true, true. 161. What is holoenzyme? Holoenzyme equals apoenzyme plus cofactor. Holoenzyme equals apoenzyme plus cofactor. Holoenzyme equals apoenzyme plus cofactor. 162. Tertiary structure of protein has hydrogen and disulfide bonds. True, true, true. 163. Complex polysaccharides are mostly homo or heteropolymers. Homopolymers, homopolymers, homopolymers. 164. Lyase are used to break CC, CX, and PN bonds. False, hydrolase is used. False, hydrolase is used. False, hydrolase is used. 165. Enzymes have primary, secondary and tertiary structures of protein. True, true, true. 166. Exothermic reactions are spontaneous and endothermic require energy. True, true, true.